For today's recording, we're going to look at one of the 5.15 specification points, and in particular we're going to be focusing upon the muscle, the skeletal muscle, and the structure of that skeletal muscle in order that we can understand how we can move from sort of what I would describe as the macrostructure right the way down to the organelle level, where we will be looking at this muscle and the fibres that make up the myofibrils, which are one of the organelles contained within the myocyte, which is a muscle fibre or muscle cell. So if we start at the top of our diagram here, we can see here that we've got somebody flexing their arm. You can see both their bicep and their tricep muscle, and from your bicep muscle, what we have done here is we have looked at one part of that muscle, which is a bundle of fibres referred to as a fascicle. So I'll just annotate that for you now. Okay, so we have the bicep, and then here we have a fascicle. Now as I just said, a fascicle is actually a bundle of muscle fibres. So you can see here that we have a collection of muscle fibres. And in actual fact, a muscle fibre is a muscle cell. And a muscle cell can also be referred to as a myocyte. So when you are reading around and you're looking at your literature in relation to this, you may well see these referred to as muscle fibres, and I believe your textbook refers to them as muscle fibres, but you might also see these referred to as muscle cells or indeed myocytes. Okay, and a myocyte is surrounded by, like any normal cell, it is surrounded by a cell membrane. But when we are looking at the cell membrane of a myocyte, this is a really quite important cell membrane, which we will refer to and is referred to as the sarcolemma. Now, a lot of these terms, if we look just briefly here, we've got a couple of prefixes that you need to be aware of. So you can see here we have the prefix sarco, and we also have the prefix myo. Okay, so these are prefixes that you are going to see quite a bit when you are looking at this topic. So you will see sarcolemma, you will see sarcoplasmic reticulum, which is used to refer to what you would have previously recognised as your endoplasmic reticulum, and that sarcoplasmic reticulum is wrapping itself around your myofibril. So your myocyte is filled with these very long tubular organelles, which are made up of your actin and myosin fibres, and we can see here we've got a myo, so again we've got that prefix myofibril, and this is an organelle, and it is a really important organelle because it's an organelle that contains the actin thin fibres and also the myosin thick fibres. So we'll just write those down, but we'll come back to those. So you've got your actin, and those are the thin fibres. And then we have got our myosin, and this is referred to as your thick fibres. And collectively, these fibres work together in order to cause a contraction. And they do this at a point referred to as a sarco. So we've got that prefix again, sarcomere. Okay, now what we need to do, so you can see here at the moment that we have got the bicep. That bicep is made up of a number of fascicles, and each fascicle is made up of a number of muscle fibres, also known as cells or myocytes. If we look at that myocyte, we can see that it is multinucleated, so that one cell, okay, so it's a very long tubular cell that we're seeing here, that one cell has got more than one, so multi-nuclei that we can see there, okay, so we can label those, so we can say it is multi-nucleated, okay, and that within that myocyte there are a number, so we've got lots and lots of myofibrils, very long tubular organelles, and those myofibrils are made up of actin and myosin fibres that are organised in a very regular organised pattern, okay, and that pattern then forms sarcomeres, where we can then see the moving and sliding of these fibres over each other in order to cause the contraction of that bicep muscle. Okay, surrounding all of these organelles, we have the sarcoplasmic reticulum, and we will look at that in detail later on when we look at the neuromuscular junctions. If we go back to what is surrounding the muscle cell, we have the sarcolemma, which we highlighted earlier. So what we now need to do is we need to look at the sarcomere in detail. So we need to take this section that is being denoted here, which is, as you can see, it's between what looks like those two black lines, and that's actually, these lines are referred to as your Z lines. And those Z lines are, we need to look at the detail between those and look at where the actin is joining at these points, and also look at then your myosin, which is the blue, thicker of the bands, which we can see between that. So as I said, where I would like to take you now is to look at the sarcomere. So that is the section that we can see in this micrograph above. So this is a short section of the myofibril, reminding you that the myofibril is an organelle which is contained within the myocyte. And a sarcomere is a short section of that myofibril, the myofibril being made up of actin and myosin fibres. And we can see that there are sort of junctions which we refer to as our Z lines that we can see here on our micrograph, and we can also see below on the picture that I have drawn for you. So we have here our Z lines. Now between that we have got a number of other bands and zones that you need to be aware of. Now, how these sometimes can be referred to are as light and dark bands that form the striations. Now, a light band is an area where there is no myosin at all, so it is only actin. So you can see in this section here, but also here. Okay, so if we're looking at the two diagrams, the micrograph, and also looking then down at the drawing, you can see a zone in which you have got no myosin, so none of the thick filament at all, only the thin actin filaments, and that is referred to as a light zone. Whereas adjacent to that, you can then see the much darker of the zones that we see on the micrograph, and that is referred to as the dark band or dark zone. So as I noted previously, the light and the dark band are made up of different filaments. So we know that the light band is only made up of the thin actin filaments, but the dark band, as you can see here, has actually got your myosin, and in some places we see an overlap between the actin and the myosin. So we can actually further break this down and look at different zones within this. So what we need to have a look at now, in order that we can understand how to recognise from a micrograph whether or not a muscle is contracted or not, is to look at those different zones. Okay, so if we start to have a look at the different zones, and we can see these here both on the micrograph and also on my drawing below, we could straight away note the M line. Okay, so that is in the centre of your sarcomere, and your sarcomere is between your two Z lines, as we can see there, and we have denoted earlier. Okay, so you can see that on the above and the below. And then the first thing that I would like to recognise here is your A band, and your A band is going to span the whole of the zone that contains your myosin, and you'll see there that particular band is going to overlap with the actin, so you can see that you've got the myosin and the actin overlapping in that area, in that A band. Okay, whereas if we looked at the H zone, that is the area that is denoted, and this will change the size of this, okay, but you can see that that is the area where you have only got the thick myosin in that space. Okay, so that is going to get bigger and smaller dependent upon whether the muscle is contracted or not. Okay, so I'm going to write in there your H zone. Okay, so that zone is going to increase and decrease in space dependent upon the level of contraction of the muscle that is taking place. Okay, so we have highlighted our A band. Now we need to have a look at the I band. Now the I band is going to sit in the space where, so we've just looked at where we've only got myosin. Now we're going to look at where we have only got actin, and you can see here in this space you have only got your actin thin filament, and this is your I band that you are seeing in that space. So we have got the M line in the centre, okay, which in my drawing below is not the easiest to see, but you can see it above in that distinct line there. And then you've got your H zone where you've only got myosin, you've got your A band where you've got the overlap between the actin and the myosin, and you've got the I band where you are seeing only actin filaments in that space. So one last thing that we might want to note here is that that space between, so you've got your Z lines where you have got it holding together those thin filaments, and we said that that forms your borders of your sarcomere. That sarcomere itself, in terms of its length, is going to vary, but at rest we are looking at approximately 2.5 micrometers, and I'm not very good at drawing micrometers, sorry, that symbol, in length, and that is at rest. And that is going to be changeable dependent on the level of contraction of your muscle at a given time. Now, in terms of the responsibility for the contraction, your A band, where we have got the crossing and the overlapping of those fibres, that's going to be responsible for that. And we need to now look into the detail in terms of how that takes place.